Hello everyone and welcome to another EAStalk webinar. We have some exciting topic to present today, and that's RAS in Research, a tool for supporting fish welfare and commercial development. Today's session will focus on the innovative work being done by Nofima, a leader in research and development in the field of aquaculture, and specifically for the role of recirculating aquaculture systems in maintaining fish health and welfare. Today we have with us Asa Espmark, Acting Research Director at Nofima, René and Anja Striberny, both research scientists at Nofima, and they will provide us with an in-depth look at Nofima's RAS infrastructures, showcase examples of research applications, and discuss how this system can support both fish welfare and industry needs. We will also take you on a virtual tour of Nofima's RAS facilities through a newly produced video. Thank you again for being here, and let's dive into exciting world of RAS technology. Asa, the floor is yours and you can start with your presentation.
Thank you very much, Anna, for this nice introduction. So, could you maybe please confirm when you see my presentation?
Yes, it's working.
Super. So once again, my name is Åsa Espmark, and I am the leading of the research group production biology in Nofima. So in this webinar, we will now put focus on research in RAS infrastructures, and we will use Nofima's comprehensive facilities as the example. You will soon see a film where Nofima RAS infrastructures is presented. We have access to 4 RAS facilities in Sunndalsøra and in Tromsø, where we mainly do research with Atlantic salmon. But we also have the possibility to do research in RAS with other species, such as cod and cleaner fish. After the film, we will highlight 2 examples of the use of the RAS facilities by my colleague Anja Striberny and René Alvestad. I do not intend to repeat or reveal what you will see in this film, but I need to emphasize that the facilities are suitable for covering a wide range of research questions, as we have both single RAS systems, where the separate systems are totally independent from each other, as you can see in this illustration here. And also the opposite, where more tanks share one RAS systems, and when there is a kind of dependency between the tanks. But these systems resembles more industrial conditions. We are proud to be part of the Aquaculture 3.0 project, where the RAS facilities in Sunndalsøra are part of the Transnational Access or TNA program, where you can apply for performing research free of charge. Please have a look at the Aquaculture 3.0 webpage, where you can read more about the TNA program and how to apply, and where you can also see what other facilities in Nofima Sunndalsøra that are part of this TNA program. If you want to see the RAS facilities in Sunndalsøra, coming to the conference Small Production in the Future, the 16th and 17th of October, is a very good idea. This biannual conference gathers R&D, industry, and governmental bodies to hear about smolt, post-smolt, and RAS. Prior to the conference, we invite pre-registered attendances for a tour in our RAS facilities. So by that, I will introduce the film and enjoy.
Thank you very much, Asa. The movie is starting now.
Resirkulering i aquaculture, eller RAS som vi forkorter det til, jo en produksjonsform som øker i Norge og verden for øvrig. Det jo mange fordeler med RAS-anlegg. Den mest åpenbarlige vel at med den høye resirkuleringsgraden så bruker et RAS-anlegg veldig lite nytt vann. Dette resulterer i at man kan produsere fisk på områder som ellers ikke så velegnet for akvakultur, på grunn av tilgangen på egnet og godt vann. I og med at et RAS-anlegg tilnærmet lik lukka, så vil vi ha muligheten til å ha full kontroll på hele miljøet som fisken lever i. Det driver og sjekker temperaturer og oksygen, at det stemmer overens. Vi tar en dobbeltsjekk av det som online-målinger mot manuelle målinger, sånn at vi sikrer at fisken går på riktig temperatur og har bra med oksygen. Det vil også potensielt være enklere å ha kontroll på smitte inn i et RAS-anlegg. Det fortsatt utfordringer og utviklingsbehov med RAS-systemer som må forskes på. Eksempler teknologisk utvikling som gjør denne systemene mindre energikrevende, og som gjør dem enda bedre tilpasset fisken og fiskens ytelse, helse og velferd. Ut av røntgenbilder av levende fisk får vi mulighet til å undersøke utvikling av nyreforkalkning. Over tid har vi utsatt dem for forskjellige påvirkninger i oppdrettsfasen. Nofima var faktisk først ute i Norge med å ha forskningsinfrastruktur på RAS. I dette bygget som vi står i nå, så har vi mer enn 50 kar av ulike størrelser og 4 RAS-systemer. Det ligner veldig et industrielt anlegg, og vi kan kjøre industrirelevant forskning, men allikevel med høy presisjon. Vi kan monitorere, måle, ha kontroll på vannmiljøet, og anlegget har stor grad av fleksibilitet. Mye av dette nybrottsarbeid, så alle som involvert i forskningen må evne til å tenke litt ut av boksen, tenke alternativt, men allikevel skjønne kompleksiteten i et RAS-anlegg. Eksempler på forskning som vi har gjort, at vi har testet ut grenseverdier for ozon og karbondioksid for laks i RAS. Vi har også testet ut ulike desinfeksjonsprotokoller for RAS, og vi jobber kontinuerlig med protokoller for å sikre smolten sin helse og velferd, og også ytelse, når vi gir fisken ulike innsatsfaktorer, sånn som fotoperiode, temperatur, ulike saliniteter, og at fisken ulikt stor når vi setter den ut i sjø. Vi har stor variasjon i den infrastrukturen vi har. For noen spørsmål så kreves det infrastruktur som ligner mest mulig på industrien sine egne forhold. Men da vil jeg likevel ha full kontroll på innsatsfaktorer og på måling og overvåking av fisk og system. Noen spørsmålsstillinger krever at forsøkene blir gjort i kar som helt uavhengige av hverandre. Derfor så har Nofima investert i enkeltrasanlegg, både på Sunndalsøra og i Tromsø. Med enkeltrasanlegg så mener vi at hvert eneste kar har sitt eget rasanlegg, og dermed så blir systemene helt uavhengige av hverandre. Eksempler på forskning som dette viktig for, for eksempel fôrforsøk. Hvis vi gir et kar en fôrkomponent, så vil vi ikke at den fôrkomponenten skal påvirke de andre karene. Så her på Sunndalsøra så har vi tilgang til enkeltrasenheter som det her. I Tromsø så de i ferd med å ferdigstille et flunkende nytt rasanlegg på havbruksstasjonen. Og her i Tromsø så har vi også tilgang på enkeltrassystemer. Her kan vi kjøre smitteforsøk, og i de systemene så har vi testet ut effekten av ulike patogener, altså virus og bakterier på rassystemet, og vi har også testet ut hvordan man kan bli kvitt de ulike patogenene ved hjelp av desinfeksjonsprotokoller. Nå står vi i det splitter nye rasanlegget i Kårvika, som vi kaller for Rasforsk. Her har vi tilgang på 24 kar og 30 karmoduler. 18 av de karene 1 kubisk kar, og 6 9 kubisk kar. Her kan vi kjøre forsøk med litt større fisk, og ettersom modulene flyttbare, så systemet veldig fleksibelt. Et resirkuleringsanlegg viktig for fremtidig produksjon av akvakultur. Det gir muligheten til lokalprodusert mat, og det store muligheter for bærekraftig produksjon for fremtidig akvakultur.
amazing video of 2 locations of RAS systems of Nofima. This is the start to the rest of the presentation. I would like to introduce Anja Striberny to start with her presentation, but I would also like to add, in case you have any questions, you can use the Q&A. After the presentations, we will start with a discussion of 15 minutes. So if there is some unclear things, or if you want to know a bit more about Nofima and the work they're doing, please feel free to use the Q&A. Anja, you can start with sharing your presentation.
Thank you very much. Can you see my presentation now?
Yes.
Great. So my name is Anja Striberny, and I'm a researcher at Nofima, at the Department of Production Biology. And I would like to present you one example of RAS research at Nofima. And this is a project called Temp Intense, which is a life cycle study of Atlantic salmon with temperature and salinity as critical test variables. And this project is led by Nofima, and it's a collaboration with researchers from the University of Bergen, University of Tromsø, University of Trondheim, and also industry partners Mowi, Espmark, and Grieg Seafood. And the project is funded by FHF, the Norwegian Seafood Research Fund, with contribution from industry. The background for this project is that, as most of you know, the Atlantic salmon is an anadromous species, and the production cycle can be divided in 3 different steps. It's starting in the hatchery with egg incubation, followed by smolt production, and then the fish are transferred to sea for grow out. And often in research, these steps are investigated separately, but there is an increasing demand to look at the whole life cycle of Atlantic salmon, because the history of the fish can impact smolt robustness and seawater performance later. And during the past years, there has been increasing concerns that we may push the salmon too much towards fast growth, especially during smolt production. So the main objective of Temp Intense is to provide recommendations for commercial Atlantic salmon production with regards to use of temperature and salinity during smolt production, and this is tested in flow-through systems and in RAS systems. And we have 5 sub-goals. Firstly, we want to document whether fish farmed using recirculation technology or flow-through perform differently. We want to assess this production at different temperatures, namely 8, 12, and 14 degrees Celsius. And we assess whether these temperatures early during smolt production affect robustness in the long term. We also compare effects of stable conditions with changing temperature on robustness, and we look whether we investigate whether gradual adaptation to salinity prior to seawater transfer prefers the salmon better to it. And finally, we have economists as part of the team who look at the profitability of intensive farming in the hatchery phase, from hatchery to slaughter. So the first part of this project was conducted at Sunndalsøra, which the research station for sustainable aquaculture, which you just saw in the movie. So we produced smolts here under controlled conditions, and then during the second part of the project, the fish are grown out by Gildeskål research station in northern Norway. During smolt production, we were utilizing 16 of those 0.5 cubic meter flow-through systems and 16 0.5 cubic meter single RAS systems. And for such a large experiment, it's not only important to have the correct infrastructure available, but evenly important is the expertise and skilled staff at the research station, which we are lucky to have. So the experimental design was as followed. We had, as I said, 4 different temperature groups. We had RAS and flow-through systems, and then we had quadruplicate tanks during smoltification. We had a 3 weeks period of salinity acclimation at the station where we transferred the fish either directly to 32 ppt salinity or gradually over a period of 3 weeks. We increased the salinity from 15 to 32 ppt, and then we follow the fish in seawater at the seawater facility. So this is just a brief timeline of the experiment. We started last year in April with 10 grams fish, and the experiment runs until November this year, when the fish are hopefully ready for slaughter at a size between 3 and 5 kilograms. And throughout the experiments, we took many samples at different time points, especially interested we were especially interested into looking at this multiplication process, meaning the pre-adaptive phase where the salmon is prepared to become a seawater ready fish, and around the salinity acclimation protocol. The fish in the experiment were all individually tagged, so we can follow the history and the growth curve of each single fish. Amongst others, samples that we took was X-rays. So this is also extremely fortunate that we have the possibility and expertise to take X-rays to study deformities of the fish during the experiment, as you also saw in the film. Water quality monitoring is an essential part of doing RAS research, and we utilized the automatic loggers of temperature, pH, and oxygen in RAS. In addition, we measured nitrate, nitrogen weekly. Total ammonia, nitrogen, and nitrite were measured routinely 3 times a week, as well as turbidity and other water quality parameters. So we are able to link water quality with fish health and performance. So we can look later at the biological samples and at the water quality. And this figure just shows an example how nitrate nitrogen developed before salinity acclimation in the different temperature groups under the 2 different salinity acclimation protocols, A and B. And all, as you can see, the values were all within the recommendations for Atlantic salmon. We also conducted seawater challenge tests to look at the smolt development, to ensure that the fish were seawater ready when they were transferred to the station at northern Norway for grow out. And you don't need to look at the details, but the main message is that that all groups, actually independent of the temperature that the fish were reared in and independent of the production system, developed good hyperosmoregulatory ability over time, and we didn't see any differences in smolt quality. Since the fish are pit-tagged, we can let them go in a common garden design in the seawater facility, and we are tracking mortality throughout the seawater phase now. And this figure just shows the mortality in the different temperature groups, both in RAS and flow-through under the different salinity acclimation protocols. And as you see here, at when you look at the number of fish, the mortality during the 2 first months after seawater transfer was extremely low. So the fish were all doing very well. But we saw a tendency that the fish from the fish that were directly transferred to a higher salinity had a tendency to have a slightly higher mortality, especially in RAS, than the fish that were gradually acclimatized to salinity. When we look at the long-term growth of our fish, we see, or we saw during the last de-lousing operation, when all the fish were measured and weighed, that the fish from the low temperature groups, independent if they were reared in RAS or in flow-through systems, were still much smaller than the fish that were reared at high temperatures. And now we're just in the process of processing all the biological samples that have been taken on the way to look at other welfare parameters, that, for example, heart health, to see if the welfare indicators differ between the temperature groups. But in terms of body weight, it looks clearly as if the fish that were reared at higher temperatures are still the growing best, that it continues also in the seawater phase. But we will have the facet first in November, when we have our final sampling. And until then, we have a little blog, which is called Lakseliv. It's until now just in Norwegian, but here you can follow the developments in the project Temp Intense. Thank you for your attention.
Thank you very much, Anja, for amazing presentation. I would like to introduce now René Alvestad. René, you can start sharing your presentation, please.
Yes, thank you. Can you see it now?
Yes, I can too. Yeah, perfect.
Yes, I will briefly introduce our new recirculating aquaculture research infrastructure at the Tromsø Aquaculture Research Station. This station is located at Kårvika in Tromsø in Norway. It's been going on for about 35 years now. It's jointly owned by Nofima and UiT, the Arctic University of Norway, but a lot of the permanent installations are owned by the Norwegian government directly through Statsbygg. The station has about 40 employees and hosts a variety of trials, up to 90 trials annually. Just to give you an idea of the geography, so we're up here in the far north of Norway. Here's the island of Tromsø, where the campus of UiT and Nofima are located. And the station is located about 40 minutes driving to the north of this. The station has considerable capabilities in terms of doing trials with fish, salmon, cod, wolffish, but also lavotropic species, both in the context of aquaculture and fisheries. So there you can see some king crab or snow crab as a part of a live storage experiment, I guess. Our facilities include culture tanks for fish, crabs, and etc. Also incubators. We have a fish pathogen facility that also mentioned, rated at BSL level 2. And this also includes a RAS facility and several flow-through facilities. There are also a number of laboratories available throughout the station. Water intake is from the sea and fresh water from a river intake. Now, the reason why I want to expand our RAS research infrastructure is one, the ability to increase the capacity of our freshwater production capacity. We have a limited freshwater supply, and RAS could allow us to increase without compromising that. Also, we're in the far north, so temperatures are very cold most of the year, and the freshwater temperatures are mostly below 1 degree Celsius at the intake. And the annual average is around 3.4 degrees. Seawater is somewhat warmer and fluctuates less throughout the year, but is still quite cold. So the ability to control temperature is quite an incentive. But also we need modern and fit-for-purpose facilities to conduct industry-relevant research and education for university students and adults. So the recirculating aquaculture industry in the Arctic is quite large. It started quite early in the late 70s through some pioneering efforts, but it didn't really catch on before, say the mid-2000s. Then these huge smolt and post-smolt production facilities were built, some of them with capacities for up to 10 million fish per year. These are major investments, maybe to the tune of 100 million euros or more. And they're also very important regional employers. These are situated in sparsely populated areas where the employment opportunities are not that great. There's also been other initiatives utilizing recirculating aquaculture in the Arctic. These are more small scale. So against this context, this new facility was initiated. We call it RASForsk. This process started in 2020. It was funded by the county government, and yeah, the sort of provision of the RAS infrastructure was put out for tender, and this was won by Alpha Aqua. There were, of course, other suppliers involved in the process, providing other aspects of the construction. There was a project group consisting of the station itself, Nofima and UIT, Alpha Aqua, and Alde Aqua. So Alpha Aqua provided an interesting setup with their nano-RAS modular systems. And these consist of a fish culture tank, in this case a 1 cubic meter tank, in this case a 9 cubic meter tank, and a water treatment module, or a box, as you see here. Also some auxiliary units like the oxygenation cone and a swirl separator here. So these water treatment units consist of a drum filter for particle removal, biofilters for nitrification, a degassing unit, a protein skimmer. We also have a heater and cooler and a UV lamp. These 1 cubic tanks have one such water treatment unit attached. The 9 cubic tanks can have 3 in parallel. We don't need all of that capacity, and we have the possibility to shift these water treatment units around. So here you see the facility and the sort of general layout. We have 6 of these 9 cubic meter systems, and we have a total of 18 of the 1 cubic meter tanks. But only 12 of them have a water treatment module attached as of now. But since we can shift these modules from the 9 cubic tanks, we can have in total 18 1 cubic meter tanks operational with RAS at one time. And this flexibility was important to us because we want to maybe introduce other components into the loop. Maybe we want to test some infrastructure for the industry. Maybe we want to include some IMTA setup. So for this kind of research we are doing, flexibility is very important. Most things can be controlled at the system level or tank level. And so we have individual control of, for example, photoperiod and salinity at the tank level. Here you see the unit processes in the nano-RAS system. So you have the drum filter here. It's small, and yeah. You also have the biofilter compartment. So there's 3 of these in series, and these can be operated both as a moving bed biofilter or as a fixed bed configuration. As of now, we go with a moving bed configuration using these Anox Calnes Biochip M biomedia. But we can change this in the future, and we can test out different configurations. Yeah, we also have this degassing plate, and below this we have a sump with a cooler and heater, and there's also a UV lamp that we can utilize for disinfection in the loop. The facility also has a conference room for giving courses and lectures, which is quite important for us. Also a lab, and there's a number of other utility rooms, control room, chemical storage, and so on. So this facility is now more or less complete, and the first activity that will go on here is this testing and education in the context of the CAND-RAS project, which is owned by UIT, led by Bjørn Steinar Sæther and funded by the Research Council of Norway. There are several partners involved: Nofima, DTU, the Freshwater Institute, BKS Nord. And the general goal of the project is to increase our capacity for education and competence building in RAS in northern Norway. But there's also a part that includes testing of the infrastructure that we have. So we've been doing some targeted performance tests of the individual unit processes throughout the summer, but now we'll also start a large-scale trial with post-smolts that will go on from now and until March next year. So that's the big test. Yeah, and that's it for me. If you have any questions after this, you can contact me by email, and you can also check out these cool websites. Thank you.
Thank you very much, René, for amazing presentation. Before we start with the Q&A, as most of you probably know, I'm the main moderator of the EAStalk webinars. And since the beginning in 2019, I was the only one who was behind the scene. But today we have a special guest with us, and that is Alistair Lane, Executive Director of the European Aquaculture Society. And he will be joining us in the discussion.
Okay, thank you. Thank you, Anna. So yeah, we have this Q&A open for you. We've got 1 or 2 questions already, but when you're talking about RAS systems in general, and certainly we wanted to do in this webinar was to present infrastructure to you so that you could see systems in other countries and what they're used for and how they're being used. So when you actually hear about an individual presentation or an individual project, it's relatively easy to think about questions that you might want to ask, and we have one of those that I'll just introduce now. In the bigger picture and in a bigger context, it becomes a little more difficult. So think really about comments or questions that you might have. Think about systems that you might have in your country or not, and put those on the Q&A, because that's a way of sharing common resources for these. And we'll come back to the idea of sharing in a little minute. We have one question here from Andre from Canada, and that's a perfect way to introduce yourself by putting that in your title, as it were, and also saying who you would like to target your question to. So we have a question for you, Anja. In the osmolarity assessment that you talked about in your protocols, was chloride and magnesium measured in blood plasma samples that you had from the gills of fish?
We measured after the seawater challenge test. We withdraw blood samples from the caudal wean from the smolts, and we measured in serum samples, not specifically taken from the gills, but from the caudal blood vessels with a Pentra analyzer. Yes.
Okay, great. Andre, I guess if that answers your question, we had a little bit of a sound issue as we were coming into you, but I think we got the picture there. So you measured it in not specifically from the gills, but in the serum, as you said, and you gave the protocol or the name of that test. So that's good. I wanted to come back also to your initial introduction that you made. And you talked about the Aquaculture project, and you talk about the transnational access. Can you just explain for us a little bit, just in very brief terms, how that works? And when we talk about transnational access, I believe it works on 2 levels. So one is actually performing research, and the other is regard to supporting services. Can you just talk a little bit through us on those 2? Thanks.
Yes, of course. Thank you for the question. So Aquaculture is an EU project. Actually, it has been 3 Aquaculture projects since 2009. And this transnational access program has grown from one project to the next. So in this last, now 3rd project, it's a very big part of the whole project. So what it means is that all partners who in this project that have infrastructure facilities available, they are offering some of the fundings through EU to be able to invite researchers to come to their facilities and perform experiments free of charge. There is a description there on the webpage of what the facilities are covering. For example, from our side, we do cover what we have here on site. So we cannot, of course, perform experiments with other species than we are allowed to introduce to the station, and also if there are special things that chemicals or feed additives or so that needs to be tested, that has also to be agreed in a way separately how to do. But everything is described there on the webpage. And then if you are interested in this, you do contact the TNA manager that is listed for all different infrastructures that is listed. For Nofima, that is Per Brunsvik. You contact him and tell him what you would like to do, and then you write an application that is very easily and described online. Besides that, and also to mention, to follow up your question, Alistair, that of course when you do come and do these experiments, you are not left on your own. There will be people here and technicians and researchers that will follow you and help you through the whole process. And the deadlines for these TNA projects are open, so you can continuously go in and apply. In addition to that, there is something called now I need to.
You're maybe thinking about the technician.
Yeah,
it's
I don't remember the exact name, but it is about technicians or other people who need training that can also apply. There is a fixed deadline that and the call is coming on the webpage, and there they can apply to come and be trained in different areas that the different facilities are offering. So here they can come and be trained in RAS. We have been having a few people coming here. They operate together with the technicians around in the facilities, and they are also free to walk around and arrange meetings with the researchers and the technicians and so on.
Okay, fine. Well, thank you for that. We have a couple of more questions that have just come in. I'm going to go now to Sarah from Italy. She had a question again for you, Anja. So at the end of the last phase of the experiment, will you analyze the internal organs to evaluate their well-being? And if so, what analysis will you do?
Thank you for that interesting question, and I'm happy that you're interested in this project, Sarah. So we will take X-rays again of the fish to look for deformities and how like the bones are developing in the different temperature groups. We will also look at the heart in terms of internal organs, because lately it's become a big problem that the fish may develop deformities. We will look at the gonads for signs of sexual maturations, and we also have a panel of different molecular methods that we will use. For example, we can do a fluidigm assay to look at the gene expression in the gill, to look at markers of the immune system. So we are planning to do a lot of assessments, and in addition, we also have the possibility to do a histological analysis of both gills and skin of the fish.
Okay, thank you very much for that. That's a lot of work that you have. You're going to present them in an Aquaculture Europe event that we have next year in Valencia in Spain. That would be great. And that's a little placement from me.
That would be great. Yes.
We have a question here from Anonymous, so we don't know who that is, but are you managing nitrate levels in the experimental RAS system? René, I'm going to ask you, because you've just described us the station that you have up north. So for you?
Yeah, managing it is, if it gets too high, we don't have many options other than increasing the water exchange. We don't usually have any denitrification capacity installed, so but I think it's mostly about designing your experiments right and doing some initial calculations and knowing what to expect, and knowing that this is in accordance with the performance and the sort of the general settings of the experiments.
Okay, great. Also, you mentioned some species. You talked about Atlantic salmon, of course, that's your main focus. And you also mentioned cod, you mentioned cleanerfish. And basically, I think that the limitation, of course, that you have in these research infrastructures is the specific licenses that you have for those species. And so therefore, you wouldn't be able to normally access Nofima installations for doing work on other species. But then, of course, that's the whole beauty of Aquaculture, because these research infrastructures exist across Europe, and therefore for different kinds of systems and for different kinds of species. You did mention, René, that up in the north as well, you're doing some work with fisheries as well as aquaculture, and there you had the example of the crabs that you have. So I guess you have specific licenses as well for species that are concerned with your part of Norway. But the question I wanted to ask either all of you or maybe to Åsa is, we've seen here two examples, one in the kind of southern part of Norway, inland, Sunndalsøra, and the other one up in the Arctic Circle. What's the complementarity between those two centres? Are they completely independent, running their own programs? How are they kind of linked? And what are the links that we might see from having those two specific infrastructures basically within the same institution?
I can start, Alistair, and then René and Anja, who both stays in the north, can fill me in. Also, first of all, from the infrastructure side, we have the capacity to run experiments with bigger fish in Tromsø. We do have 100 cubic tanks also in Sunndalsøra, but I think that the new bigger tanks that they have in Tromsø are more suitable and modern to run experiments with larger fish. And we know also that land-based slaughter is becoming more and more common, and research questions regarding how to allow people to how to allow fish to stay on land their whole life. What is the effect of that, and how can we improve the fish health and welfare to allow them to stay on land? But then, of course, outside the facility by itself, it is very interesting that we have a south and north gradient there, where we are facing climate changes and we are facing increasing temperatures in the sea. And you mentioned cod. It's a very cold water species, and there are industry ongoing where they want to put cod also in the recirculation facility. So and we are doing research on climate changes. So I think in that sense it is very complementary that we have facilities in this gradient. And even though Sunndalsøra is not in the southern part of Norway, it is still in the area where the water temperatures during especially summer and autumn can be quite high.
René, Anja, do you want to come in on that, or has Åsa done a great job for you?
I have nothing to add.
No, I think having several facilities also enables us to have some different experiences gained from the different stations, and this can be shared. We can, become better together and sort of have a diverse portfolio of experiments, and this increases our knowledge of operation, and it's really just a benefit, I think.
And that also kind of partially answered a question that I had for you, Anja, which is, in your experiment, you were looking at a transfer of about smolt weight of 60 to 150 grams. And I was going to ask you about the potential for larger smolt, of course, because when also talked about the effects of climate change, also questions about health and parasites and welfare, it seems to be that the trend is either going towards fully on land for the whole cycle, or at least to try to make the seawater cycle as short as possible. So the variables that you have with regard to the economy and the cost of production may be more controllable, let's say. So was the constraint that you had on smolt size in your particular experiment limited by the infrastructure?
It was basically limited by the original call of the funding agency, actually, because they wanted that we focus on classic smolt production and not large smolt production. So post-smolt production was somehow ruled out by the call, but definitely this would be very interesting to look at different parameters during large smolt production under controlled conditions. So I I wouldn't say it was limited by the by the infrastructure. It was basically just not the topic that the funding agency in this case was interested in. But we are involved in several large smolt projects, both René and I, led by Trine Ytresdal, a colleague who is working from Sunndalsøra, and there we are going to dive deeper into large post-smolt production and production conditions during post-smolt production and the long-term effects after they've been transferred to seawater and how they perform there, given different production protocols. But there we will not be able to do any controlled experiments, but we will look at production data, which will be made available by industry partners.
Thank you for that. And of course, as we come back to the original words of also in the introduction, we're focusing on basically as a tool for health and welfare, specifically research. So that's great. We're coming towards the end of the time we have allocated to the webinar. We've got 2 quick questions, so they probably can have quick answers. First one we have from Krishna in Spain. It's about the Transnational Access Program, and he's asking about its applicability for PhD students, not just for early stage researchers. Maybe
it is open for everybody, old, young, PhD or student or researcher. It is, but it is shorter term experiments. So it is not possible to fund a whole PhD, to put it in that way. But you can, if you have a scholarship, if you are a PhD student, you're very much welcome to come and apply for experiments that you can use in your thesis.
So a final question, as we say, we're coming towards the end of our time now. Samuel from Italy has a question about the possibility of running IMTA, so multi-species experiments in the facilities. We talked about licenses for the different species that are required, but for example, in the Tromsø infrastructure, can you run multiple species or IMTA experiments, or are the tanks basically designed for single species trials?
Granted that we have the appropriate licenses, we can do this, I think, in principle. Technically, there's no limitation on this, or well, there's some limitation, but we can facilitate a lot of different configuration in this respect. Yeah, it would take some creativity and refitting, but it's possible.
That's what I'd like to hear. So nothing's impossible, and that's a very good way, I think, to end this Q&A. I'd like to thank you for your participation in that. I'd like to thank our audience for having given us some food for thought through your questions, and so many thanks for that. I'm going to hand that back now to Anja so she can do a wrap up of the webinar, and I'd like just to express my thanks for having been with us. Thank you.
As Alistair said, thank you very much for joining us today for another EAStalk webinar. We hope you enjoyed it. This was, as I said, quite a different approach to the webinars we are doing usually with a video and with another moderator. This webinar is recorded, and once everything is edited and uploaded on a YouTube channel, I will send you an email. So thank you very much for the attention, and hope to see you soon next month.